In this presentation, we'll overview autism spectrum disorder beginning in fetal development. Let's get into it. In the first trimester, neural progenitor cells, specifically the radial glia, rapidly divide in the ventricular zones of the developing brain. New excitatory neurons then migrate along these glial scaffolds to the cortical plate. This process is guided by signaling proteins like renin, which helps organize the brain into proper layers, while axons and dendrites extend under the control of growth factors and adhesion molecules. Several autism-linked genes like RELM, CNTNAP2, and SHANK3 are active at this stage, showing how early genetic and molecular influences shape later circuitry. At this stage, development can be disrupted by gene variants, maternal immune activation, severe oxygen deprivation, infections, or toxic exposure. Postpartum studies of autistic brains often show patches of disorganized cortical layering, which strongly suggests that these disruptions occur before birth. The result is a brain that forms slightly miscalibrated microcircuit templates even before the major wave of the synapse formation begins. In early life, the brain undergoes massive synapse production followed by activity-dependent pruning. In autism, research shows evidence of early brain overgrowth during the first 1 to 2 years, altered spine density, and disrupted pruning or autophagy signaling in some subtypes. The typical pattern that emerges is excess local connectivity combined with weakened long-range connectivity, especially in integration network. Long-range circuits are white matter pathways that connect distant brain regions, including frontal parietal, temporal frontal, and frontal limbic networks. These pathways also form the core hubs of the default mode network. Long-range circuits support planning, social meaning, perspective taking, language, and executive functioning. In autism, these circuits are commonly underconnected, which limits large-scale information integration. Local circuits are short-range, densely packed microcircuits within a single brain region, such as early visual or somatosensory cortices. On average, autistic brains show an excess of local synapses due to underpruning, leading to heightened gamma activity, local hyperconnectivity, and exceptionally strong detail-oriented processing. When long-range networks are only mildly disrupted, individuals may show savant-like traits with preserved or enhanced intelligence in specific domains. When long-range networks are severely disrupted, this is more likely to result in an intellectual disability. These outcomes fall under the same diagnosis, but they reflect very different wiring patterns. When local networks become overactive, sensory hypersensitivity or hyperdetail processing emerge. Local circuits can dominate over integration networks, producing intense special interest and repetitive behavior. Again, the diagnosis may be the same, but the balance between local and global processing determines the behavioral profile. Monotropism describes a pattern where attentional resources become narrowly focused. Because short-range networks are highly active, attention becomes locked onto a limited set of interests, often with extreme intensity. The dominance of local circuits also contributes to repetitive behaviors and reduced cognitive flexibility. The core issue is once again the imbalance between local and global processing systems. The default mode network is a long-range system involved in self-referential thought and mind-wandering. In autism, studies commonly show reduced connectivity within the default mode network and between the DMN and other networks, though patterns vary by age and method. A useful shorthand is that the DMN tends to be noisier and less synchronized across long distances, which affects self-processing and social cognition. Maternal immune activation refers to fever and inflammatory states during pregnancy. Large population studies show that these conditions modestly increase the odds of autism, although the overall population risk remains low. Mechanistically, evidence from animal models used cautiously to inform human inference points to IL-6 and IL-17A signaling, microglial priming, and altered cortical development. The key practical takeaway is that treating fever and infection during pregnancy matters because untreated fever itself poses a biological risk. This slide shows what happens when a pregnant mother's immune system is artificially activated, usually with a viral mimic like poly-IC or a real infection. This triggers a strong inflammatory response where we see cytokines like IL-6 and IL-17A signaling increase alongside alterations in the mother's gut microbiome. The offspring of these MIA mothers show changes in brain development and later display autism-like behaviors. By contrast, control offspring from mothers that did not undergo immune activation show typical neurotypical behavior. The key takeaway here is that maternal inflammation during pregnancy can program long-term changes in the offspring's brain and behavior. The core biological chain begins with maternal inflammation, signaling IL-6 and IL-17A, priming the microglial cells to alter cortical development. This diagram features the MIA cellular mechanism and rescue pathway. When a mother is infected, antigens activate her immune system and shift T-cell balance toward pro-inflammatory Th17 cells while reducing regulatory balance. Cytokines like IL-6 cross the placenta and activate fetal astrocytes and microglia, producing neural inflammation and neural damage in the developing brain. That pathway is associated with ASD-like outcomes. The lower panel shows the rescue mechanism where regulatory T cells release IL-10, suppressing inflammation, reducing IL-6 signaling, and restoring normal neural growth. This demonstrates how immune balance is the critical factor in shaping fetal brain outcomes. Here's the full sequence collapsed into one model to get some perspective. Early in development, small errors in layering, migration, or synapse assembly create subtle miswiring. During the synaptogenic boom, these early templates get amplified, producing excess local branches to compensate for their weaker long-range structure. Pruning then becomes biased. Weaker signals persist locally, while long-range refinement is reduced. The final macro pattern is local sensory and associative overgrowth with weakened integration hubs, especially the default mode and frontotemporal systems. Acetaminophen is metabolized into a toxic byproduct called NAPQI, which is detoxified by glutathione at normal doses. Only overdose depletes glutathione. While autism does show signs of oxidative stress and lower average glutathione levels, which led to concern about prenatal acetaminophen exposure, causality is unproven and confounding from illness and genetics is substantial. As of 2025, professional guidance remains unchanged. The ACOG reaffirms appropriate use in pregnancy, WHO classifies the evidence as inconsistent, and the FDA now uses cautious language due to mixed data. Vaccines remain biologically and epidemiologically unrelated to autism. People with autism often show elevated oxidative stress and lower than average blood levels of glutathione. Several nutrients help replenish glutathione, including selenium, vitamin C and E, alpha-lipoic acid, whey protein, and amino acids. N-acetylcysteine, or NAC, is particularly important because it supplies cysteine, the rate-limiting building block for glutathione. NAC also directly scavenges for free radicals and reactive oxygen species, reducing cellular oxidative damage. NAC affects two major signaling systems: the nuclear factor kappa beta system and the nuclear factor 2 system. The nuclear factor kappa beta system drives inflammatory cytokines like IL-6 and IL-1 beta. When nuclear factor kappa beta is highly active, it suppresses antioxidant gene expression by inhibiting NRF2. NAC indirectly dampens this inflammatory loop. NAC actually supports the NRF2 pathway itself, which activates antioxidant and detoxification genes. By boosting NRF2 activity, NAC increases cytoprotective enzymes and reduces oxidative stress, which in turn lowers inflammatory signaling. Interventions work best when they match the biology. Early behavioral and developmental therapies leverage brain plasticity to strengthen long-range cross-domain networks. Sensory-aware environments reduce background noise so that integration networks don't become overloaded. Language and social scaffolding explicitly teach inference, perspective taking, and flexible switching skills that rely on long-range connectivity. Medical stability is also critical because sleep, seizures, GI issues, ADHD, and anxiety all degrade network coherence. Reducing oxidative stress, including through agents like NAC, when clinically appropriate, helps protect neural tissue from damage. Pushing long-range integration demands too early, such as unstructured group discussions in noisy environments, reinforces avoidance rather than growth. Misreading autistic communication as defiance leads to punitive responses that increase stress and impair learning. Masking mandates, which force continual performance of neurotypical behavior, lead to burnout and reduced functioning. The correct approach is supporting authentic communication instead of enforcing constant compensation. Autism is fundamentally fundamentally a neurodevelopmental timing and circuitry condition that often begins prenatally. Many different genetic and environmental pathways converge on the same macro pattern of local overconnectivity paired with long-range underconnectivity, with timing and subtypes shaping the final profile. Interventions are most effective when they reduce sensory noise, stabilize physiology, and explicitly train integration rather than only targeting the surface behaviors.